So building on what Ed talked to you about in his presentation. So, thinking about your proposal, we wanted to give you some background into what makes a good proposal. And so talking about NIH guidelines, really is the starting point. So what Ed described really is that each proposal has to argue strongly that that idea has significance, it has feasibility, it has a good approach to answer the question, there has to be innovation. So focusing in on your challenge for your candidacy exam that will be coming up, this upcoming academic year, we're really not going to focus too much on investigator because that's not really the point of the exercise. Environment is the same for all of you. So again, not the point of the exercise. So what I want to talk about in terms of your proposal idea for your candidacy exam is the kind of three main criteria that Ed talked about for you. One is significance, the other is feasibility, and then the third is going to be innovation. But to be honest with you, innovation is the hardest one at an early career stage like yours. So we're going to focus a lot more today and this whole class really on the issues of significance and feasibility. And by the way, feasibility, I really mean like a solid approach. Is it going to work, right? Do you have methods that will work and they will answer the question? So does that make sense? So we're going to really focus on those criteria. Ed, can you just confirm that you guys are seeing my screen?
Yes, please, you can see.
Can you see my screen now?
Yes, please.
Yes, we can see your screen and we can hear you very well. I'm sorry, I was muted. I don't see any raised hands, so in case if I will see, I will.
Okay, yeah, just let me know. We're going to be quite interactive today. We're going to doing some literature searches. So, okay, so like I've been doing my last few, before we get into the nitty gritty of significance and methods and feasibility, I wanted to go through a few myths because I feel like I want to make sure you guys are clear that the challenge we're asking you to do here is pretty tough, but I want to dispel some preconceived notions. And one of them is that, oh, okay, I just read 3 papers for my homework assignment 2, and I have lots of new ideas. I don't have to read any more. I don't need to read that many papers. I just need one idea. And I would say that that's a misconception. Really, the best ideas come when you read a lot of papers, you consider a lot of different ideas, and you pick your favorite. The one that when you got that idea after that paper, it really made you excited. Okay. So our advice, and the reason that we're having you do this again for homework 3, is that you really want to conceive of lots of different ideas so that you can have lots to choose from. So you can pick really the best one. So conceiving of many ideas based on many papers will ultimately allow you to identify one idea that's your favorite and worthy of pursuing in your research proposal. And so you should really be reading and conceiving of as many ideas as you can. And so to help encourage you to do that, homework assignment number 3, I'll put it early here, is to do more or less the same thing you did for homework assignment 2. So the same thing you did for today's project, today's assignment. In this case, so again, you're going to read 3 papers, reverse outline them and kind of summarize them, and then generate new ideas. But in this case, the difference is that these papers have to be different than any paper you've read so far. They can include a review paper, but we're not requiring it this time. So, but 2 of the papers, at least of these papers, excuse me, have to be, so by the way, I'm in the area right here. At least 2 of the papers have to be primary research papers, but all 3 of them can be this time. Only 1 review paper if you like reviews. But in this case, all papers must be identified on your own. You need to find your favorite journal and find papers there. You can do some keyword searches on topics that you find interesting, COVID-19 or I don't know, I'm just making stuff up. Okay, so please find 3 new papers and get new ideas, because we really believe that the more ideas you have, the more likely you are to have a great idea. And I think I just saw a hand raised. If you want to unmute yourself. ask your question.
Yeah, so I was gonna ask, since you said the 3 papers will be different from the ones we had, do you mean like in a different field, or you mean like it can be in the same field but different from the one we did?
Yeah, it can be in the same field. I mean, as long as it's distinct from your research advisors. Remember that the clear requirement of your candidacy exam independent research proposal is that it's distinct, different from your research advisor. So as long, I mean, it can be in like, you know, if your research advisor is doing medicinal chemistry of protein X, you can still do medicinal chemistry, but it has to be of a completely different system. So it can be in the general area, but it has to be distinct. You can pick a new field if you want to. It's up to you, but you can stay in the same general area. If you did, if you found 3 papers in medicinal chemistry for assignment number 2, you can do medicinal chemistry in assignment number 3.
Okay, thank you very much.
Good, thanks for the question. Great. Okay, so so continuing on, this assignment is going to work the same way. Summarize, come up with your ideas, and then submit to homework 3 assignment. Okay, any more questions about homework 3? Okay. All right, it should work similarly, but find more ideas. Get some really nice ideas that you can feel you know, excited about. Find that one idea that really makes you excited. Okay, so let me dispel one more myth before we move into our activities. The second myth is the idea I conceive of today is my final idea, and I will use it in my independent research proposal. And the fact is that's not true. All ideas evolve and change, and they kind of take on kind of new and improved elements. So the fact is the idea you have today may be kind of the foundation or the shell of the idea that becomes your proposal. But please let the idea grow and evolve and become amazing through the process of reading, okay? So please, you know, please allow your idea to grow. Okay, allow your idea to change. And that's really when our best ideas come, is when we let them sort of become great, okay? So how do we get to that point where we make our idea great? Okay, so that's what we're gonna talk about today. How do I, so our topic is, how do I assess the quality of my idea? How do I vet my idea? And so the fact is, the way you do that is by reading more and more and more, okay? But in this case, instead of the reading that we've been doing up to this point, and for homework 1 and 2, has been kind of reading lots of different papers, maybe in the same area, but kind of confronting different challenges in that field, right? So getting lots of different ideas from different areas. In the case of assessing the quality of one idea, you really want to focus and read strategically to explore specific questions related to that research idea, okay? And the questions that you're going to explore strategically in this reading, in this type of reading, are going to relate to significance and to approach or feasibility. So some of the significance questions, building on what Ed just talked about, what about my idea is significant? Is it in a significant field of study? Is it really going to impact human health? Is it going to impact quality of life? How is this, what's the field I'm in? And is it a significant field? Is it really going to do something to improve human existence in some way? Does my research idea represent a significant problem in that field? So let's say that I choose to work on something really significant, like new batteries to power our devices, right? But if the problem I identified doesn't confront that important area directly and significantly, then I'm not going to make that progress. You need to make sure that your idea is not just in an interesting area, but addresses a key problem that is confronting that area. And then finally, I think a great way to kind of vet whether your idea is significant is, if I'm successful with this idea, what will happen to the field? Will it expand enormously? Will something become possible that wasn't possible before? So if I'm going to make a new battery and this is my idea, will the new idea result in like a huge leap in the kind of battery technology that we can use? Does that make sense? So those are the ideas you want to be thinking about in terms of your idea. Now let's talk about the approach, or what I really like to call feasibility, because when you're designing experiments to test your idea, your hypothesis for your science, you want to make sure your idea is testable. So is the idea I have, is it new? I mean, has anybody done this before? Is my idea really a new idea? Or has it been already done? Now, there's a positive and negative there. If it hasn't been done, that's great, because you could do it. But if it has been done, you have to say to yourself, well, that was a stinking great idea. There's some, if you find that one of your ideas has already been done, you should pat yourself on the back and say it was a really good idea. Congratulations. But you got to find one that actually, you can work on. Another issue of feasibility is, can I test this idea? Are there experiments that I can design that will allow for me to decide whether my idea is working? Okay. If there are no appropriate experiments, maybe you can design a new method, which would get you to that idea. But the point is, you have to have methods, experiments that you can propose to test your idea or your hypothesis. Finally, I think something that's really underappreciated for proposals, especially proposals like yours, where you can't do experiments to really test them before you submit your proposal. Something that's underappreciated is that you can look in the literature and see if anybody else has done anything kind of related to what you are proposing to do. Have people use the method that I'm proposing to you, and if they have, use their experimentals to learn about how you would interpret the data and get the data. If nobody's used the method, then look at other aspects. Has somebody used something like my method? Has anybody proposed to a hypothesis that's kind of like mine, so I can show that my hypothesis really does have feasibility? Okay, so looking at prior precedent for your idea is really a great way to show that it will work. Okay. So I want to focus then on these two issues of significance and feasibility. I know what you're saying is, I haven't hit innovation. I will do that a little bit later, because innovation is the hardest one to do. I'll hit that in a little bit. So what I thought we would do together, maybe for the next 10, I don't know, 15 minutes, was to really start vetting an idea together. I want you guys to understand that what we're doing here is something that you can do by yourself anytime, okay? Because I'll show you how we can do this together. You can do this. I know it means a lot of reading, but it's really going to be a fun process. So what I thought I would do is go back to our discussion last class period, where we came up with some novel ideas based on this paper we read together about viral induced self-assembly of magnetic nanoparticles. So if you remember, at the end of class last time, together we came up with a few ideas. And I thought this idea about COVID-19 detection that one of you guys submitted was really cool. So I thought, let's together assess the quality of this idea, okay? So let's do this together. So we've decided that we think COVID-19 detection with these magnetic nanoparticles could be the idea that we want to pursue. And let me say that what we're about to do together here, you should be thinking about doing with your best one, maybe best two ideas, okay? You can't do this with every idea you have. You're just going to have too many of them. So pick the one that feels the best, and you would do the same thing. Okay, so I'm excited. When I came up with this idea of a new COVID-19 detection using these magnetic nanoparticles, something in me got really excited. I want to see how good it is. So I want to ask you guys a question. I want us to be a little more interactive now. My research idea is, the solution is do magnetic nanoparticle detections of COVID-19. That's my solution. But I have to decide first if there's a problem or a gap in the field that needs this detection, right? I actually don't, based on the paper we read, I have no idea. Is COVID-19 detection a really big problem in the field? So why don't you guys, so let's come up, sorry, I have to get out of my thing here. So let's think about kind of ideas about how we would go into the literature and explore the gap in the field. So what do you think is a problem that this detection method could be solving? So I want to bring that, I want to say that, I want you guys to give me some feedback there. So you can either write in the chat or raise your hand and Ed will call on you. So why don't you tell me your ideas here? What do you think could be a gap in the field that is solved by this detection method that we've thought of together? Oh, my computer's not working. Do you guys need me to start off? I waited my 7 seconds.
We don't see any raised hands.
Any raised hands? Right, so some of the things that can cost and availability of test. So what we would need to do there is we would need to read the literature on already available. So we would need to learn about already available COVID-19 tests, right? Are they good?
The thing that I'm gonna add here, since you're asking the whole class, is, you know, how do we really need to know in how good or bad of a shape the subject really is?
You mean the patient?
The patient. So that's definitely that's potentially an unmet need, because, you know, you're treating a patient, you think it's going well, and then for some reason patient dies. Why does that happen, right?
So maybe I just got right. So Ed, your point was who can be tested, right?
That's a good interpretation.
What type of patient can be tested? Okay. In the chat, I see another question was, you know, speed. How quickly can the test be implemented? So maybe that's related to your point, Ed, right? So speed of implementation, right? Do you have to be in a hospital to get this method? Can you just do it at home, right? Home tests would be cool. Let's see, what else do I have? Yeah, so how quickly? Right, so the sensitivity, another question was how much circulating virus is needed for that to be detected? So we need to know the sensitivity.
Right, and one other comment that I'm seeing here in the chat is reliability, I'm sorry.
Yeah, reliability.
This, yeah.
How reproducible. So, and remember, so the sensitivity of test, and so these things really relate to the population. So if I do this, in terms of reproducibility, if I do this test 10 times, will I get the same answer, right? Okay, cool.
Another point that I'm seeing is specificity to the particular type of virus. Do we really need to have an antibody? That's the question that that is being raised.
Well, in this case, we'll be detecting the viral particle itself. So we won't be, this is not an antibody test, right?
Exactly.
Right, so will this be specific to COVID-19 and not just any virus, right? Oh, right, and so another comment was, could this test be used to determine how critical the health is of the patient? I'm not sure about that. That's pretty serious. Okay, so these are great questions that we're asking ourselves. So is there a gap? So remember, we don't know yet. Is there a gap in the field? Like, are the COVID tests available now? What kind of costs are they? Could we maybe improve on cost? Can they only detect virus in certain kinds of patients? Could our tests be more sensitive? Could it be more reproducible? Could it be more specific than current tests? Does that make sense? Right, another question is, how safe is the method? How easily can we do that? Okay, these are great questions. And guess what? We can answer them with literature searches. Okay, what about, let's talk then about the methods, because now we need to be able, so let's say that we decide, after we do a literature search, we decide that it's significant, and we'll do this literature search together. What kind of methods do we think we want to use? So I'll give you guys a minute to write down. So what kind of method should we go back to? Should we go back to our paper, and we can see the kind of methods they used were light scattering over here. They had atomic force microscopy, but most of their experiments were done using this thing called a relaxometer.
And I see one comment from Kate saying non-invasive sample detection.
Yeah, okay, so right, so that should go up here. I don't know why this thing won't let me. There we go. Okay, so non-invasive, thank you.
Well, maybe that's related more to methods question.
Right. So can we figure out if ours will work without something, without being invasive, right? So I was thinking here, actually, what experiments would we do? Sorry, I don't know why I just made that in red. I'll turn it, there you go. So a non-invasive, right? So if we're trying to test our method, we want to make sure it's not invasive in a patient, right? What else? We need a method to just determine the sensitivity, the reproducibility. So we need to find like a way of assessing the aggregation of our viral particles. So can we just maybe use this relaxometer technology that was in the original paper? Should we maybe think about that? Right, so another student said use MRI, and Jordan's really excited about the spectrodyne. So I'll put that down because I have no idea what that is, Jordan. So maybe we need to find out what it is together. And actually, this just shows you, like if Jordan was my peer mentor, he would give me this idea and maybe send me on a great direction. So thank you, Jordan, for being my peer mentor. I see Jordan in the chat that it's a particle sizers, right? So it could see aggregation of the viruses compared to non-aggregated. Yeah, cool. So maybe we should look at that. Okay, so anyway, I think the point here is that Jordan's been really excited about this idea. I kind of am too, I'll be honest with you. Okay, so now that we have some questions, now this is what you do, is you have an idea, you start thinking about the details of it. Is it solving an important problem? Is it in an important area? I think we all know that COVID-19 is an important area, so I guess I won't argue with that here. But is it an important area? Do I have methods that I can use to assess this idea? So, so I thought we would do is just Google search. This is what Ed talked about last time, is a level 1 type search where we're just going to Google search something. So I thought would be cool is we can just look to see, because honestly, a lot of the research on COVID-19 is kind of new to the, I mean, it's so new that we may not have everything in sort of traditional journals. So I thought maybe we could just search for COVID detection methods and see what we find. And I did this earlier today, but every time I have a feeling that it's changed in the few hours that I did this. So let's see. So we can see here that we get a good RX for coronavirus testing, which is interesting. Testing for COVID, this is CDC. I think that would be pretty good resource, right? I see Wikipedia, and Ed talked about last time, Wikipedia is a great first place. You cannot use it as sort of like one of your references in your proposal, but it's maybe a really good place to get some general knowledge. Here's a protocol for detection using CRISPR diagnostics. So that sounds to me like a very new way of diagnosing CRISPR. And then here's a paper on, a review paper on diagnosing COVID-19. So what do you guys think of those? Like, what would you spend your time on here? I mean, in my opinion, I kind of think about, I would maybe start with the Wikipedia page just to learn something. Yeah, so Wikipedia is a good place to start. But then maybe going to this actual peer-reviewed review paper would be a good place to start. So if we go to the Wikipedia page, let's just real quick look through it together. COVID-19 testing can identify the SARS-CoV-2 virus and includes a method to detect the presence of the virus itself. RT-PCR, isothermonucleic acid amplification. So actually, through Wikipedia, if you don't know that much about PCR or this amplification method, you could learn about it, right? And I did look at this before, so I did look at this site before. So, you know, it gives you sort of a very basic overview of PCR detection. Oh, look, you've got some nice pictures. There's a PCR machine. You know, so Wikipedia is maybe a good first start. And in some cases, actually, you know, Wikipedia does have at the very bottom, which is way down here, it does have some articles that you can look up that could be peer reviewed. So Wikipedia is a good place to start. Let's go back and let's look at instead this, this article that's in the PubMed website, and it's actually been published in ACS Nano. So in this case, it's a review article, which will go through some of these same parameters. This review article, you could actually cite in your proposal, and this would be actually a better, a more reliable, you know, reliable source. Okay. That's cool. Okay, so does that help a little bit? Let's do something else. How about another thing that I think would be really useful here? So if we go back to our ideas, these review articles could tell us things about cost, sensitivity, reproducibility. And my understanding about just reading the newspaper is that reproducibility is actually a really big problem. That may be something that our assay could really improve upon. So I think that would be a great place to find real data to show it. Okay, let's move on. Another thing that I think is really helpful that I would strongly consider you doing is what's called a forward search. So what I would love to know is who has been working in this magnetic nanoparticle field, and has anybody else done virus detection using these magnetic nanoparticles? Because if they have, that is actually proof that my idea could work, right? Or our idea could work. Sorry, I'm starting to get possessive of this idea. I think it's a great idea. Okay. So what I can do is what's called a forward search. So what I'm actually going to use for this, I'm going to use Google Scholar, because I actually think it's a really good resource. I know that it's publicly available and all of that, but I actually really like it for this. So what I'm going to do is actually just take the information from my paper. So this is, in our case, it's our paper we read together, but in your case, it's your paper that you read and you were inspired with an idea. And you can see that I found my paper. Okay, what you're going to be looking at is the cited by. This is the number of not just papers, but also kind of recently come out information that have cited this, this paper. In addition, you can see that citations, the forward search, so anything that cited this paper in Web of Science, which, you know, Web of Science, Ed went through 3 sources: Google Scholar, PubMed, and Web of Science, right? Web of Science is reviewed, high quality articles. This cited by in Google Scholar, you can see that the cited by in Google Scholar is many, many more papers cite this paper. And that's probably because they're not all peer reviewed, right? So if we go, I'm going to pick the Web of Science 400 just to see, you know, what has cited. So this is a forward search. This paper was published back in whatever 2018 or 2016. I don't remember what time. When was it published? Oh, 2003, a long time ago. So lots of papers have cited it now coming forward. That's why we call it a forward search. 400 papers, but I don't want to read all these 400 papers. I'm going to refine my search to only looking at those papers that say something about a virus. So here we go. So these are all the papers where I can look at viruses. Scientific advances in diagnosis and emerging viral pathogens. Okay, that might be a nice review article for me to read. Quantitative detection of biological nanoparticles. No, that doesn't say anything about viruses. Review on viruses on protein assembly. I don't really care about protein assembly. I want to see detection, although I guess it's related. Maybe if I need more background, I can go back to that one. Like fluoroimmunomagnetic detection of rotavirus. Okay, here's one about rotavirus. Let's see, anything else? So I like so you might, oh, but that's in plasma, in blood, or in, you know, a sample. So anyway, the point is that you can see, you know, oh, here, maybe recent advances in nanoparticle concentration and their application to viral detection. Okay. So these give you ideas of what's been done with this nanoparticle technology related to viruses. Oh, look at, this one just caught my eye. Potential of magnetic nanoparticles for hepatitis, for hepatitis B detection. That sounds like the paper I want to read. How do they do it? How sensitive is it? How cost effective is it? And that way I can learn about the method that I'm developing here. Okay, so I guess I might want to stop there. How do you guys feel about what we've gone through? I mean, we really, when you're thinking about your idea, you want to be very critical of it and try to come up with ways of deciding whether your idea is really a good one. Okay, here I am. Okay, any questions before I move on? I'm almost done.
No raised hands.
Okay, so another area that I just want to mention, if I have a few minutes, is that when I was reading my paper, this paper, which, you know, as I mentioned to you earlier, magnetic nanoparticles are not in my specialty. I do not know that much about this field. So when I was reading the paper, I wrote down little things that I didn't understand. So for example, I've never performed a light scattering or an AFM or a relaxometer experiment. So I decided, you know, I told myself that if I was to pursue this idea, I'd probably want to learn about those things. How would I learn about those things? Well, I would do what we did and just search for it, right? Other questions I had down here were, what's the sensitivity of the PCR-based method? Is mine going to be better? How sensitive does the viral detection assay have to be? These are some of the questions we came up with ourselves. So as you read your papers, be thinking about these forward questions so that you can be prepared. This is how you decide on your best idea that you want to spend time really thinking about and developing and coming up with idea, you know, further validating for significance and feasibility. Okay. Okay, good. So chime in if you have any questions, guys. In the end, you know, we're really just trying to make this understandable and relatable so that you can do this yourself. So if there's no questions about understanding the issues of significance, am I doing something significant to the field, and you know, the method and how feasible, and it solve my question, I'll move on to innovation. So last call on questions about significance and feasibility, and how to do searches, and how to find papers that will let you think about that. Okay, so let's move on to innovation. So innovation is the hardest, because innovation is really, has anybody done things like I've proposed here before? And I would recommend, I think as you get older and you're better, you really have read a lot, you can be assessing innovation as you go, but I think at your early career stage, I would worry about innovation last. And the reason is because I feel like you can assess the innovation of your ideas after you've spent some time thinking about the significance and the methods and the feasibility. So, you know, while you're reading about significance and feasibility, ask yourself a few questions. Have others explored a similar hypothesis? And if the hypothesis that you're exploring, in our case with our example, you know, magnetic nanoparticle identification of COVID, you know, there's probably some parts of that hypothesis that have been done. Probably nanoparticles have been used for other viruses. And other detection methods are COVID. But has anybody put magnetic nanoparticles with COVID? That's innovation, right? Have there been similar systems tested? So, you know, if you're doing a medicinal chemistry problem, have them in other inhibitors of your protein already been created? If not, there's innovation. Are you using similar methods? Let's say that you propose to develop a reaction and you're going to use a combinatorial array of ligands and catalysts to develop your method. And that would be using, you know, kind of a combinatorial library of reactions. Has anybody done that for the reaction you're talking about? So your choice of method could be innovative. So as you're reading, think about what's been done by the field, and maybe if that hasn't been done by anybody, that could be a source of innovation. Okay, so my point is that you really want to assess the feasibility and significance of your idea and keep an eye out for innovation. Don't make it a driving force at this point. Ed and I need to make it a driving force perhaps in our research, but you know, we're at a later career stage, and so you know, don't really worry about this too much at your career stage. So my point is that at your early career stage, you know, innovation may not be the driving criteria. Let significance and feasibility be your driving criteria, and then innovation is something you can follow you know, later. And the reason I think for your candidacy exam as a second-year graduate student next year, your thesis committee is going to be the least critical if there isn't a huge amount of innovation. They will, though, need to see significance and feasibility, no doubt, okay? And hopefully, as you grow as a scientist, the issue of innovation will be something that you could start contributing to. That's the idea. You can become a PI. Okay, so last parting thoughts I have for you, papers to find ideas. As you read about the significant feasibility of your favorite idea, write everything down, okay? I cannot tell you how many times I got pulled into a rabbit hole in the internet and just kept reading lots of things and I wasn't writing anything down, and then I couldn't remember where I saw that. Don't do that. So let yourself go down the rabbit hole, but write down how you got there, because you need to know how to get out. As I mentioned earlier, please don't focus on too many ideas for this vetting process. Maybe pick the one idea that you got super excited about when you had it. Maybe two, but honestly, one would be better if you really like one, because you just don't have enough time to explore every idea. This is something that even experienced scientists, we can't explore every idea. Just go with the ones that you think are really exciting. So I said go with your gut. Which is the ones that really made you excited? You're going to be spending a lot of time on this idea, so you better be excited about it, right? And then finally, you know, I think another question I would have is, you know, when do I know that I've done enough vetting to pick my favorite idea? Like, how do I know that I'm ready to make that into a proposal? I guess my advice here is that, you know, if you can answer the issues, if you can say, yes, my idea is significant, and yes, my idea is feasible, you can do a proposal on it, okay? The innovation, again, can come afterwards as you read more and more and more. And please realize that you will be reading more and more and more and more papers as you write this proposal. If you think that you're only going to be looking for literature in the next week until you have an idea, that is wrong. You're going to be reading more and more and more literature, because as you write, you're going to realize, oh, geez, I guess I don't know how I'm going to do that. I got to read something to figure out how that method is going to work. So writing, learning, reading, letting that, remember how we talked about letting the idea grow and mature and become its own amazing, great thing. That's what you have to let your idea do. Okay. So I just want to end by giving you a sneak peek of what we're going to do next time. At our next class, we will start the process of writing. So we really want you by next class to have some idea of your favorite idea. Now, the good news is you might say to yourself, wait a second, I have so much to do. I need to read a lot more papers. I need to find I don't have my idea today. Ah, okay, don't freak out. If you're not ready for that yet, it's okay, because remember, Memorial Day is next week on Monday. We will not meet on Monday. So you actually have until next Thursday, so a good 10 days from now, to read, get your idea, your favorite idea, vet the idea, decide on the significance, feasibility, and then be able to write about it by the 28th. So you really have enough time to do this. So hopefully we've shown you how. That's our job, is to show you how. Now take the time to really make. Okay, folks, I'm going to stop there. Any last questions about what we talked about today? The issues of significance, feasibility, innovation last. How do I look for papers to answer those questions?
No raised hands.
No? Okay. So with that, then I think what we'll do is move to our breakout sessions. I emailed you, I'm sorry I didn't email you earlier. Some of the peer mentors suggested that I email about the breakout session. So I did that, but I'll try to if I'm gonna email you in the future, I'll try to do that a little earlier. But in any case, what you're gonna do in your breakout sessions tonight is to go student by student, each of you, to discuss one of your 3 papers you talked about that you worked on for your assignment, and the favorite idea that you have. So you want to think about, what was my favorite idea of all the things that I talked about, that I learned about, and talk about that paper and your favorite idea. And hopefully now, after you've presented, so take about 2 to 3 minutes to discuss the paper and the idea. So what was the field, what was the gap or the problem, how was it solved, what were the conclusions, and how did you come up with your idea, okay? So take about 2 to 3 minutes to talk about that with your group. Then for another about 2 minutes, I want you guys to be thinking about questions that that student could pursue in a literature search to decide on significance and feasibility, okay? So if you hear something that you say, oh yeah, you better look at this problem because I worry that that might be already done. Bring up some ideas, help each other, you know, kind of brainstorm together. Remember, for the candidacy exam, you guys are allowed to talk to any other graduate student. You just can't talk to faculty. So help each other. All right, folks, thanks for listening in. I hope we've given you some guidance on what the next steps are. Have a wonderful breakout